Hello students, in previous session we have completed transcription process. Transcription is the process in which the genetic information is transferred from DNA template, not the entire DNA, a part of DNA so called gene, towards the RNA, so that all three RNAs, messenger, ribosomal, transfer RNAs, they code for amino acids. They help in polymerization of amino acids on the ribosomal units to synthesize protein, because protein serve as a regulator hormone. Protein act as a biocatalyst enzyme to complete metabolic reactions in the cell to generate the product essential for the cell. The product of transcription is RNA molecule. In today's session, we are learning types of RNA molecules. RNAs, except some viruses, in rest of the cells, we call them as non-genetic because they are not heritable. But they will get the entire information for sequencing, polymerizing, and the length of the protein from the DNA molecule. There are three types of RNAs depending upon their location, depending upon their function and the structure. Here, from the left hand side, the most important RNA molecule having shortest lifespan. Compared to tRNA and rRNA, they just forms only 5%. Shortest means lengthwise, according to the number of nucleotides present in it. And shortest means soon after synthesis, it undergo translation, and after protein synthesis, mRNA degenerates. It will not remain for a longer time in the cellular space. Why we call messenger RNA? As it carries coded information for polymerization of amino acids into proteins from the gene. A set of genes working together. 2 days back, I completed transcription unit, which is composed of 3 types of genes. Promoter, who initiate the synthesis of RNA production, and structural region, who actually give coded information in the form of triplets, and termination region, where the RNA synthesis stops. So transcription unit is a group of genes. It is also known as operon, so that we are learning in next few sessions. Operon is a set of genes working together to synthesize RNA molecule, and that process we call transcription. And for transcription, the catalyst required is RNA polymerase. As we have already learnt, in prokaryotic cells, there is only one RNA polymerase available. It may be production of any RNA, but in eukaryotes, there is a polymorphism in catalytic activity. During production of mRNA, RNA polymerase II will bind to promoter. If there is a synthesis of tRNA, RNA polymerase III will catalyze the entire process. And for the production of rRNA, RNA polymerase I is essential. Let us start learning the structure of different types of RNAs. Messenger RNA is a straight molecule, single-stranded, having polarity from 5' to 3'. There are total 7 regions I have shown on this messenger RNA molecule. It is prokaryotic messenger RNA. Just recall in previous session, when I explained transcription in eukaryotes, after splicing, it is the transformation of hnRNA into functional messenger RNA, where introns are cleaved and exons are brought together. Soon after splicing, there is a capping occur where methylated bases are added and tailing occur. Adenylated bases are added. So the number of nucleotides added towards 5' and 3' end are representing the 5' end, and 3' is the tail region. Next to 5' end or 5' end, the second region is leader sequence. Approximately 300 to 400 nucleotides are available. So when messenger RNA is released through nuclear pore, it has to move towards specific ribosomal unit. So leader sequence will guide messenger RNA to reach the respective ribosomal subunits. When ribosomal subunit attaches, it will see that region number 3. So binding site for ribosome is initiator region. If you read the sequence of nitrogenous bases there, these are ribonucleotides: adenosine monophosphate, uridine monophosphate, and guanosine monophosphate. It is initiator codon. Codon is a sequence of 3 nucleotides. So AUG is always initiator codon. You take any messenger RNA from the universe. It may be from prokaryotic or eukaryotic, from unicellular or multicellular, any type of cell you take. Messenger RNA is having initiator codon AUG. Sometimes it is GUG. This initiator codon is always coding for a amino acid methionine. So first amino acid present in any protein synthesized by the cell is methionine. It is a initiator amino acid. From that amino acid, further amino acids start linking with each other by peptide bonds. So AUG is initiator region from where protein synthesis, so called translation, begins. Then next to initiator, fourth region is sensible region. Here there are number of nucleotides available. It depends on the requirement of the cell. Here triplets are continuously arranged in the sensible region. So depending upon the demand of the cell, here number of triplet codons are available. They are continuously arranged without any punctuation. They never overlap. They are always universal, means a set of 3 nucleotides is specifying single amino acid during protein synthesis. So there are many triplets available here, and it varies from one mRNA to other mRNA. So I have not written anything there. It is the functional region during translation, or length of the protein depends on this sensible region. Total number of amino acids in a protein except initiator depends on sensible region. Sensible, they are sensing the amino acids. Clear? Region number 5 is terminator region. So here I have written triplet codon UAA. It will not code for any amino acid. Some mRNAs are with UAG. Some mRNAs are with UGA. So these 3 will not code for any amino acid. Hence we call them as nonsense codons. Okay, though they are nonsense, but they signify a very important role in protein synthesis, because the translation process is inhibited by non-coding any amino acid near terminator region. So these 3 are terminator codons. To remember this sequence without any confusion, right, I always refer to Marathi words, Ala and Gela. U stands for Uttam. Okay, why I have taken Uttam? It is not my favorite name. There is one sincere person working in biology department as our tender, right? So I consider his name to remember. So to recognize these terminator codons and to remember lifetime, consider UAA, it is Uttam Ala Ala. Next, UAG, Uttam Ala Gela, and UGA, Uttam Gela Ala. So these are the nonsense things to remember the terminator codons. Clear? There is no confusion at all, because in most of the competitive exams they will ask you you should identify the terminator codons from the given multiple options. After terminator region, there is a trailer region. It will help the mRNA to release from the ribosomal units once the protein synthesis gets over. And the last seventh region is poly A tail, representing 3' end. Thus, messenger RNA, which carries coded information with initiator region, continue with sensible region, and terminate with UAA region. Let us move to the second RNA. It is called transfer RNA or tRNA. Robert Holley in the year 1968 proposed clover leaf model to learn structure of tRNA, because these are the longest tRNA molecules and total 20 types of tRNAs are suspended freely in soluble form in the cytoplasm. Right? So remember the location of tRNA, cytoplasm. Multiple tRNAs are available. They are transferred because they are carrying activated amino acid towards ribosomes according to the triplets exposed on the messenger RNA. As I said, it is highly folded molecule showing different loops. The loops are formed due to accumulation of unwanted non-coding nucleotides, right? So as usual, the tRNA is having 5' to 3' polynucleotide. Because of foldings, some regions are brought together in anti-parallel direction. So wherever anti-parallelity is found, there we can see the base pairing between adenine uridine, between guanine cytosine, cytosine guanine. There is no thymine here, uridine and adenine. Like this base pairing, it is temporary, not constant like DNA molecules. So this diagram carry 5 marks weightage. These are the labelings essential when you draw the diagram. It is called adapter molecule because it serves various functions. You can see the loop here towards my left hand side. It is DHU loop, dihydroxyuridine loop. This name depending upon the nucleotides present there. Now what this loop will do? It will help in activating required amino acid. So how it will come to know that this is the amino acid necessary for protein synthesis now? It depends on the activity of anticodon loop. Here more than 200 nucleotides are available. Out of them, 3 will recognize triplet codon on mRNA. So codon of mRNA recognized by complementary anticodon of tRNA. Or in other words, codon and anticodon should be complementary. So when codon is exposed on the ribosomal unit in mRNA, then a specific tRNA having complementary anticodon gets activated and it will specify DHU loop to activate amino acid. For example, initiator codon on mRNA is AUG. Then what will be the anticodon? I said complementary. A's complementary is U, U's complementary is A, and G's complementary is C. So what is anticodon? UAC. So when AUG is exposed, tRNA with UAC will pick up methionine. Activated methionine is added to CCA end. This is 3' end where triplets are available CCA. They will take or activated, they will help in attachment of activated amino acid, and that's why we call this as amino acid binding site. Then tRNA with functional active amino acid is known as loaded or charged tRNA. This tRNA should move from the cytoplasm towards ribosomes. They are usually attached to the surface of endoplasmic reticulum. For the movement or going in a specific direction, who will help? It is psi-U loop. It is pseudo-uridine loop. Clear? So single molecule serving various functions. That's why we call tRNA is an adapter molecule. Function of tRNA: recognizing codon with the help of anticodon. Function of tRNA: activate amino acid. Third function, to load that amino acid, and fourth function, to carry that amino acid. So it works like a vehicle sending required raw material towards the site, and site is ribosome. Clear? Coming to the last RNA molecule, it is ribosomal RNA. It is abundantly found RNA in the cell associated with the proteins forming the units, and these are nothing but ribosomes. So ribosomes are formed by rRNA with proteins. Okay, so more than 80% RNA in the cell are in the form of rRNA. There is no specific structure studied here, but these are polynucleotides packed with the proteins, okay, showing the ribosomal units. Now what is the duty of rRNA? As they forms the ribosomal RNA, they will receive required messenger RNA, decode the triplets one by one, allow loaded tRNA with amino acid, and help the polymerization of amino acids by developing peptide or amide bond. This is function of rRNA. So in the cell, it may be prokaryotic or eukaryotic. During translation is nothing but polymerization of amino acids into protein. So transcription, synthesis of RNA, which will help in translation, which is nothing but synthesis of protein. So based on the function, there are three names given: messenger, transfer, and ribosomal. With this, I will conclude this session. Hope it is very clear to you, right? mRNA structure is not important for board exam, but you should know the knowledge. It is, you can say, it is the proper structure. Without this, there is no protein synthesis you can follow further, right? tRNA structure is important. Clear? So we can compare 3 RNAs and try to remember the important terms. Thank you very much.